welcome everyone to Science Society, and of course, a special welcome to you, Ophir and David. It's so nice of you to, take the time to come here, go through the hassle to create the account and everything. We really appreciate it. And now we figured everything out. So let me start with introducing David Castillo Zufeifa. I hope I said it more or less right. Uh,
Perfect.
he is the principal scientist and group leader at Genentech in South San Francisco, California. And do you know by any chance Rosa Barreda?
Ooh, I don't. It doesn't ring a bell, but
She's an
it's quite a
immunologist.
big. Yeah, it's okay. I'm in immunology in immunology, so I should might
Oh,
have
but
crossed paths with her. Yeah.
yeah, because she is also at Gentech, principal scientist now, and yeah, I thought maybe she was in
Oh,
my PhD group class, so that's why.
oh,
Yeah. Um. So yeah. So David, he went to a university at the Universidad de Costa Rica for his bachelor in biology, and then later to the University of South Carolina, a professional science master in biotechnology, and he did then later his PhD at the University of Colorado Anschutz in cell biology and stem cells and development. Um, yeah, and then we are very honored to have here Professor Ophir Klein. He is a professor at Sinai and the vice dean at the Children's Services at Cedars Sinai. And he is a David and Meredith Kaplan Distinguished Chair at the Children's Health Cedars Sinai, and he did his bachelor at the University of California in Berkeley, and then later his PhD in genetics at Yale University, and his MD also at Yale University School of Medicine. Um, and before he was assistant professor at the University of California, and he held various really impressive positions before being here at Genentech. So it's an honor to have you here, and thank you so much. And yeah, as I said a few minutes ago, we start with the question: how did you discover that you wanted to be a scientist or work, in research? And yeah, we said that David would start. So thank you.
yeah. Thanks, Katarina. Thanks for having both of us. So I should say that growing in a tropical country, rough, like tropical Costa Rica, it was it was quite easy, I believe, to get into biology because you see so many things, so many like high diversity of plants and animals around you that it's actually not an untypical dream of people back home to think about biology. So I think that definitely growing there that helped. However, what I should uh, come clean about it is that my path from biology to what I'm doing now hasn't been that um, let's say straightforward. It has been more I would say going with the flow and with uh, as time passes, learning a little bit more and more about different things that biology uh, encompass. And I remember that after my undergraduate studies in Costa Rica and being in South Carolina, one of the I would say defining moments was the paper from Shinja Yamanaka, where they uh, discovered these iPS cells. Like, how can we basically go from a somatic differentiated cell into revert them into a stem cell? And that was like a key moment for me when I was like, how is this possible? How can people do this? And that's where I would say my passion for stem cells it started. And as during my PhD, like that was like also a defining moment, figuring out not only how we can make new stem cells, but actually that already in the body we have stem cells that we can harness and we can kind of manipulate and drive them into um, different types of tissue that would benefit patients in general in the future. And yeah, and ending up in Fierce Lab as a postdoc, that's where my passion for intestinal biology started, and that's what I'm doing, continuing my research in my lab at Genentech now.
Well, that's wonderful, and it's interesting to hear. Yeah, it's very nice to hear that this environment that you grew up in kind of as enforced you to become a scientist and work, be curious about your environment. That's really nice, and I think yeah, it's really important for our children to just be outside and and experience the world and not just their iPad. So please, parents out there, listen to this.
Totally agree. Totally agree.
Yeah, that's a great story to support that thesis. So, and yeah, Ophir, is it would you like to
Yeah,
share your story?
sure.
Thank you.
Yeah, no, mine is a little bit more serendipitous than David. So actually, when I started, I can't say that I was uh, obsessed with biology when I was growing up, and maybe it's because I didn't get to grow up in a beautiful place like Costa Rica, more in suburban California. But I started university and actually completed my degree in Spanish literature. So I was kind of a late bloomer when it came to science. But I was fortunate began when I began in college. There was a graduate student who lived in my dorm, who was also my TA for chemistry, and there was an opportunity to work in a lab. And she said, "Oh, you should give this a try." And I just found that I really liked being in the lab, and so I then went through a number of different thought processes, and decided I was going to go to medical school, and then I did an MD PhD, and there I really spending a lot of time in the lab. I found that I really liked it, and I did my PhD in viral genetics, um, and clinically became very interested in clinical genetics and then developmental biology, which is related. So then decided I was going to study organ development, and through developmental biology, then got into stem cell biology. So it was a little bit of a winding path to where I ended up.
Yeah, that's nice because then you got to learn a lot of different topics and see problems from very different angles. It's really interesting. That's also an interesting story because I got into neuroscience because I like uh, electrophysiology. I was really struggling I had to decide where to do my master's and did the rotations, and I was kind of starting neuroautonomy. Oh my God. Like, I was like bored, and then I was electrophysiology, and I, oh my God, okay, I found what I like to do. I'm so glad. Yeah, that's nice. And yeah, from there, Ophir, how did you kind of alluded already how you got into this field, but maybe is there a story, like a peek behind the curtain, how this project came about? Like, was it, was pretty straightforward? Were you about to give up?
Yeah,
Um,
no, actually,
no.
unstraightforward. I can tell the first half, and then I'll pass it along to David, who actually did all of the work on it. So our lab became interested in epithelial stem cells in general over a decade ago, and through a collaboration with a colleague at Genentech, where David now works, we got very interested in the gut and had been looking at various cellular behaviors, primarily how stem cells enable renewal and questions around identity and plasticity. And I would actually point you to a nice review that David wrote with another former postdoc on some of these questions that he published a couple years ago. And I had a friend at UCSF, this guy Todd Neistol, who was working in Drosophila and was interested in this gene called DLG1 that he thought was very important in stem cell competition in the fly. And he had a postdoc who began to look at whether the same thing was true in the mouse. And they began to work with a mouse model. And then the postdoc left. She got a faculty position. And Todd said, well, maybe you guys want to help us sort of wrap this thing up. We have a lot of data. You know, we have promising hints that maybe it's involved in cell competition, but not clear yet. And so I had a former postdoc in our lab who I asked to take this on, and she began to look at it. And it wasn't totally clear how strong the signal was for cell competition, but she had a hint that there was something interesting going on. And then she ended up actually getting recruited to go to biotech, and so the project was a little bit abandoned again. At that point, I talked to David and Tom Wald, another postdoc in the lab, and asked them if they would be interested in looking at this. And they did this amazing job of taking a story where there wasn't really a strong sense of what was going on and making what I think are some pretty remarkable discoveries. And so at this point, I'll turn it over to David, since he was the one who actually was there day to day doing this. I'll let him tell the very winding path that this story took. I think it's the most circuitous, at least one of the most circuitous stories that we've had in the lab.
Yeah, yeah, it was first off, I think like I want to reiterate how important it is to talk to your colleagues. Well, obviously in science that it's super important because like really this collaboration was, as Sophia said, it started between him and Todd, and really Todd comes from a Drosophila a background, and Ophir having mainly focused on mammalian biology. It was very a very in, uh, actually joyful collaboration. But again, it started just with a simple conversation between colleagues, and yeah, like as Sophia was saying, it has been how there are some stories that just click, right? You start doing an experiment, and it's kind of a like a snowball. It starts to get bigger, bigger, and things just start fitting into place like a little puzzle. But definitely, this was not the case with this story. What, which makes it, I think like it definitely holds a dear place in my heart because of that. And one of the key things that I learned from this process, and I still it's sticking in my mind, is like uh, after every lab meeting that we had, Tom and myself presenting our updates of fear, I would say like most, like I would say like 99% of at the end of each lab meeting was like, guys, great job, keep going. I know it's we're getting closer and closer, and perseverance will pay. And that really has stuck with me because this is really a story where perseverance was something that we had to learn. Um, so so yeah, as also Theo was mentioning, the story started with really beautiful data from Todd's lab. Um, However, as we started, when Tom and myself started uh, digging deeper into the biology in the mammalian intestine, we started seeing a different phenotype arising, and a phenotype that we had to, what I think was to be creative, creative to in designing experiments to really shine a light on the on this phenotype, because initially it wasn't that obvious. So what I can just conclude by saying is that it was definitely, I remember also in one lab meeting we had um, this little picture of the game Life. I don't know if you guys played that game growing up. That is like you go through twists and turns as you grow up. You well, some people buy a house, buy a car, more responsibilities, and there's all these twists and turns that you progress slowly through life. And I remember that we showed Tom and myself just walking through these uh, intricate game, and it really mimicked our journey in this project. That again, with all the complications that it had, I think that made it uh, a more joyful challenge and project.
That's interesting to hear. It really also, the cost was really high, so you value it more. But
Mm-hmm.
I'm so glad that yeah, that you kept going. That's really wonderful work, and we get to discuss it also now here too. So, yeah, congratulations, and yeah, we are now very curious to hear your presentation. And everyone, the slides are pinned on top of the room. Feel free to access them. And David, yeah, the stage is yours. Thank you.
Yeah, sure. Thanks. So I would also uh, tell viewer that please chime in on also your perspective as we develop and as you remember the key conversations as
Yeah, for
we were
sure.
putting this
You're,
story together.
you're doing a
Uh,
great. So but I'll chime in.
Perfect. So one we started. So as we mentioned, we started this project in a different uh, with a different mindset initially. As we progress at analyzing the data, we got um, interested slowly on this question: how do stem cells die or regenerate? On this very very big picture question. And if we go to the next slide, um, what these the reason why we got interested in this question, and we are uh, both of Fear's lab and my lab, the reason why we're interested in stem cells and how we what are the key mechanisms that keep the stem cells happy, basically, that simply put, is that if we understand those mechanisms, we can um, in the future, or well, I would say right now, we can actually understand the mechanisms of going from a healthy tissue to a damaged, and then what is more important, going back to a healthy tissue. And we believe that as developmental biologists, uh, we believe that the stem cells are really at the center of these of this process. So if we move to the next slide, the after thinking about the overarching question, okay, what how stem cells, what do stem cells require to survive? What we know is that in homeostasis, these cells are the workhorses that drive the renewal process. However, these cells rely on microenvironmental signals that are coming from what we call the niche. So the niche is composed of other cell types that provide very simply put, provide all the goodies, the nutrients, or well, I should say, all the proteins required for the stem cells to be happy. Interestingly, these niche signals that the stem cells receive are not static. For example, the level of activity of a specific signal can fluctuate between the states of homeostasis and regeneration, and the stem cells are required to properly interpret such fluctuations. And this is where understanding that how stem cells know how to respond to the signaling pathways. This key question is the one that comes back to how do stem cells die or regenerate. So to us, this question of interpreting fluctuating signals by the stem cells was not clear, and that's where we got quite interested in exploring this question in the small intestine. So if we go to the next slide, you'll see that the small intestinal epithelium that is depicted here by this picture, it's a beautiful array of villi and crypt structures. And this epithelium is a multifunctional tissue that participates in absorption of nutrients, and very importantly, it maintains a barrier against the external environment. In doing so, it is under constant damage, so it must rapidly repair and regenerate to maintain tissue integrity. And what is quite surprising is that the entire epithelium can renew in 3 to 5 days, making it an attractive model to study somatic stem cells. And again, ask this question of how these actively dividing cells respond to dynamic niche signals. So if we look at a cross section of the intestine, what we'll see is that it's composed of a single layer epithelium. We have in the small intestine finger-like protrusions called villi, where the majority of nutrient absorption takes place. And then we also have a small epithelial invaginations into the mesenchyme that are called crypts. And these crypts are the ones that house these workhorses that we call stem cells. These stem cells that we identify them by a specific marker called LGR5 are the ones that give rise to the different cell types that compose the intestinal epithelium. So from this point on, we're going to focus on those tiny epithelial invaginations that we call crypts. So if we go to the next slide, what is depicted on the left, that is on the illustration under homeostasis, is the intricate signals that come from the niche. that are required for a stem cell self-renewal and maintenance. And one of the main signaling pathways that you can see that is depicted in the illustration is the Wnt signaling pathway. We have Wnt ligands coming from panet cells that are interspersed between the green LGR5 positive stem cells. We also have other Wnt ligands, as well as a Wnt modulator called R-spondin, coming from mesenchymal cells that are enwrapping, that are around the intestinal crypts. What we know is that during injury, for example, in this case depicted here, a rotavirus infection, or also during irradiation, or in inflammatory diseases like inflammatory bowel diseases, we know that during these processes of injury and followed by regeneration, there's increased Wnt signaling. So we know that is essential for the process of regeneration. However, too much Wnt is not okay. If we tilt the balance to overactivation of the pathway or aberrant constitutive activation of a Wnt, this can lead to cancer in the intestine, colorectal cancer. So if we go to the next slide, this is where, as I was mentioning, talking with colleagues, talking to your neighboring labs, talking after conferences in happy hours, it's not only fun, but really it is where I believe the best science takes place. And the reason is that we got interested in this protein, this large one, or DLG1 for short, thanks to our collaborator, Todd Nistel at UCSF. DLG1 is part of the scribble polarity complex and is required for cell polarity in different epithelial cells, and is conserved from Drosophila as well as in mammals. And the reason why we got interested in it is Todd pointed out to us the fact that DLG1 can interact with a key member of the Wnt signaling pathway, that is APC. So he knew that we were interested in Wnt signaling. This, as we mentioned before, this project started at looking at cell competition, but we kind of converge in this collaboration, both two interests. Our interest, Tom, the other postdoc in Ophir's lab, and myself, in Wnt signaling and DLG1. Again, thanks to the collaboration with the conversations with Todd. So now we knew that we needed, if we wanted to pursue these, we needed to first, very simple, look at the DLG1 expression in the intestine. Is it actually there? Is it something to look at or not? And what you can see in the next slide is that DLG1, depicted in green, is basolarly localized throughout the entire epithelium, that is in the crypts as well as in the villus region. We can use genetic tools where we can genetically ablate, get rid of the DLG1 gene, in this case under a stem cell driver, LGR5 CRER, and after 14 days of chase, let's say after 14 days of inducing DLG1 loss, we see that we lose protein expression also in the entire epithelium. So we were very excited about this. We were like, okay, we're starting to cook now. We have a protein of interest that it might be a key player in one of our favorite signaling pathways. Our genetic tools are in place, they're working. Let's see what happens. Well, this is the first turn in our life journey in this story. What we found is that when we get rid of this protein, nothing happens in the intestinal epithelium. And this was puzzling to us because it has been widely documented. It has been very broadly documented that DLG1 is required for cell polarity. So we thought that we were going to actually disturb, at least at some level, the intestinal epithelium and its organization, and that wasn't the case. So we had to go back to the drawing board, and what we realized, looking at the literature, is that in our first pass of reading papers, we didn't notice that most of the previous correlation between Wnt signaling and DLG1 function has been seen in high Wnt conditions, meaning in a context where there is overabundance or activation of the Wnt pathway. For example, it has been shown that loss of DLG1 decreased survival of APC mutant mice where the Wnt canonical pathway is hyperactivated. Also, it has been shown that in patients with Crohn's disease that experience chronic inflammation, there's lower DLG1 expression, and in these conditions, there can be increased Wnt signaling, and it has been reported that loss of DLG1 in the intestinal epithelium may be involved in the pathogenesis of this disease. So if we go to the next slide, knowing this, we knew that we needed to challenge the intestine. And when I say challenge, I mean that we needed to design an experiment that we could increase Wnt signaling, but at the same time, leaving the creep compartment intact, because we were interested in, at the end of the day, we were interested in the stem cells itself and the creep compartment. So we couldn't injure the crypts because that's what we were after. So thanks to an awesome collaboration with Mary Estes' group at Baylor, we were able to use this very elegant injury model that they have developed that is infecting mice with rotavirus. And the reason why this is such a, in my opinion, such a beautiful system is that the rotavirus infects villus tip cells, differentiated cells, and promotes a cell death of those cells, as you can see in the lower panel in the rotavirus infected villus that we are looking them from the top. There's increased cleave caspase 3 marker for apoptosis in cells that are at the top of the villus. Also, what is very interesting about this system is that the creep compartment is completely spared from injury. But what they have reported previously is that after infection, there's increased Wnt signaling in the intestinal niche. So with this system, we were getting the best of, let's say, two worlds. We were keeping our crypts intact, and at the same time, in a physiological relevant model, we were able to increase Wnt signaling in our DLG1 knockout mice. And we had an aha moment here where we saw that when we look at whole mount crypts from the bottom, we start seeing these beautiful blobs of dying cells inside the crypt compartment. And needless to say, as you can see in the top panel, this is a very rare event that we would to find in wild type conditions. So we knew, okay, we are now getting this phenotype, and it aligns to what has been published, that it's possible that DLG1 requirement uh, uh, is attached to high Wnt conditions. So if we go to the next slide, this is just quantification of the phenotype, where we quantified the number of crypts that contain at least one dying cell. These we did all this in whole mount tissue. Then the other quantification is that we see a slight, still significant decrease in the number of total crypts in the small intestine. So if we go to the next slide, what we decided to do next was to take advantage of the organoid culture system. We can culture intestinal mini guts that are intestinal organoids on a dish in vitro, and the benefit of doing this is that we can titrate, or we can take out factors that are required for organoid growth, and one of those factors is Wnt ligands. So basically, we can reduce Wnt signaling or increase Wnt signaling, and by doing so, we can assess the impact of these fluctuations in Wnt signaling in the organoid system. The other key advantage of the organoid system is that it's composed entirely of intestinal epithelial cells. So we can directly test if this is an effect on the intestinal epithelium. And also, we can enrich for stem cells and ask if this is a stem cell dependent phenotype. So after explaining all that and the benefits of using this in vitro model, what we first did was to culture intestinal organoids in what we call a standard Wnt media. This is what it would be equivalent to homeostatic Wnt levels. And what we found is that slowly, however progressively and steadily, we lost DLG1 knockout intestinal organoids. However, this loss took about 47 days, so it was quite a slow process. Now, when we increase Wnt signaling by adding exogenous Wnt in the media, we can speed up this process of losing the organoids by a matter of hours. Now we start losing these organoids. They start to form these very small structures and condensates that you see in the bottom panel by as quickly as 24 hours after induction, after adding Wnt media. And there's even more of these condensates by 72 hours. So we quantify these. We see that there's definitely a drop in the formation of these organoids. Then, if you go to the next slide, these are two videos. I don't know if you would, if everyone would be able to play them in their computers, but what you would see is that we have a control organoid on the left and a DLG knockout organoid in the right. And when we expose organoids to these high wind conditions, we know that we enrich for stem cells, and we get these balloon spheroid-like structures, organoids. And what is labeled in magenta is the epithelial cells, and what you see in green are cells that are undergoing apoptosis. And what you can see, hopefully, is that in the DLG knockout spheroids, they completely collapse, and there is a higher accumulation of dying cells. So what was interesting, and to us quite interesting, is that high wind conditions that are seen in regeneration are now beneficial. The system requires these, actually now become detrimental after the loss of this protein called DLG1. And we know that this is a requirement, at least in the stem cells, because we know that in this in vitro culture system, the majority of the cells that compose these spheroids are the stem cells. So if you go to the next slide, we then decided or wanted to know what might be going on downstream of DLG1. And for this, we took an unbiased approach, and we decided to isolate DLG1 knockout intestinal stem cells that express LGR5 GFP. So we can FACS sort GFP positive cells that we know are stem cells. And after isolating them at 2 days or 14 days after these cells have lost DLG1, we did RNA-seq. And usually when we take these unbiased approaches, as many of you do, we are expecting to get an amount of targets that we can then explore, and basically we can even start an entire lab and several PhD projects based on a single RNA sequencing experiment. However, what was again interesting, as this whole story was to us, is that we found just one gene that was differentially expressed, specifically downregulated between wild type stem cells and DLG knockout stem cells. When I say that it was the only gene that was differentially expressed, I mean that it was the only gene that was consistently at day 2 and day 14 differentially expressed. However, we get other genes that are differentially expressed either at day 2 or at day 14. But since this gene was consistent, we decided to focus on it and try to dig deeper on its function. And this gene is called RGAP31 and is a CDC42 GTPase activating protein. So if you go to the next slide, you'll see that we validated our RNA-seq experiments via a single molecule in situ hybridization. We see that RGAP31 is indeed expressed in the intestinal epithelium. After DLG1 knockout, we lose expression transcripts of RGAP31. If you go to the next slide, and I'm almost at the end of the presentation, what we what we decided, or well, let's say we were actually lucky to find someone to collaborate with us on exploring the function of ARGAP31, and this was also this was a collaboration with Natalie Lamarche Bain at McGill. At this point, we contacted her. We told her about our story, and she got quite interested in it, since her lab works on these GTPase proteins, and she had in her colony ARGAP31 conditionally knockout mice. So we imported these mice, and we used our most robust assay, that is the formation of these organoids in vitro in the presence of wind, and tested if ARGAP31 could phenocopy the loss of DLG1. And well, that was a long explanation just to something that you are seeing very clearly in these images, that there is indeed loss of the of intestinal organoids as we expose them to high wind. So if you go to the next slide, what you will see is that first off, on your left, there is a quantification of active CDC42. As I mentioned before, ARGAP31 is a GTPase protein that can and will deactivate the function of CDC42. So our prediction was, well, if there's reduced ARGAP31, this should lead to an activation of CDC42 in the intestinal epithelium. And when we measure active CDC42, that is what we saw. And what was way more interesting, and at this point, this is where things started to fit into place after years of work. We found out that CDC42 is actually a downstream activator of the Wnt planar cell polarity pathway, and this pathway is involved in cell proliferation and cell migration, basically cytoskeletal dependent phenotypes. So one of in conversations with Natalie, with one of the key experiments to test CDC42 function is cell migration. So we decided to culture these 3D organoids into 2D monolayers and treat them with a high wind. And hopefully you can see, if you play the videos, that when we do a wound assay just by scratching in the middle of a confluent dish with intestinal stem cells after wind signaling, you can see that the DLG knockout organoids are not able to close the wound, and they take quite longer to move the same distance as the control to the organoids. So I would just wrap up here with a summary that what we found is that downstream of wind signaling, we require DLG1, and downstream of DLG1, we have ARGAP31 and CDC42, and this molecular axis is essential in intestinal stem cells for two things. Basically, what we found is, one is proliferation as well as cell migration. And what is quite interesting about this story is that it tells us that something, that having too much of something is good, but in the right context. If we in this case, if we lose DLG1, too much of something good actually becomes becomes bad, or let's say, quote unquote, toxic. And yeah, the last slide is these take home messages that I already covered them. So um, I won't go through them, and and this has been really a tour de force. Like, it really takes a village to push this project forward. All of it was done under the guidance and the the pep talks from Ophir to persevere. And I should say also, all these my partner in crime was Thomas Wall, and other collaborators within UCSF, as well as other collaborators outside UCSF. So yeah, that's what I had to share today, a snippet of the story. And yeah, we'd love to hear if people have any questions or comments.
Yeah, David, I just want to say you did a great job with that. So thanks for putting that whole thing together.
Oh yeah, for sure.
Yeah, thank you so much for sharing this really impressive work. And again, I'm so glad yeah, you moved you continued moving along, because it's really important work to see that balance. And I feel like you see that in all kinds of different areas of, it reminds me a little bit, for example, of cortisol in the brain, like during development, if you have like stress factors, it can be that you end up with too much or too little, and both is kind of bad for mental health type of resilience. So it's really interesting to see that that balance in the right context is so important. On the other hand, don't you feel sometimes like, oh my God, all of this is so fine-tuned. Will we ever be able to like fix something? How do you feel about that?
Ophir, I don't know if you. I would just say that type of questions haunt me, and at the same time challenges me to keep doing these. I think that yeah, biology is so perfect uh, and so complex that well, I'm thankful for that, because that's why we have we have a job. and
Yeah.
um, and but yeah, it's really haunting and sometimes overwhelming, right? How these fine uh, balance is maintained, and is it, can we actually get to uh, uh, understand it to the fine grain detail, to be able to manipulate it for the benefit of patients. I think we can, and there has, well, there has been already uh, uh, very seminal um, therapies that do so, but definitely it's challenging.
Yeah, thank you for that. And then the follow-up question I have is, did you start looking into, I know that you want to, I guess in the future you want to go into, human cells samples probably to analyze this. Um, do you see in different, or do you expect in different disease models this balance to be kind of widespread, this equilibrium being out of balance in different cell types and not just in the intestine? I was thinking, for example, there was a news article this week out that fertility in men is going down, and probably it is related to pollution and so on. Could a similar system be responsible for various kind of pollution related, stress factor disorders, basically.
Huh. Interesting. I hadn't thought about that. I don't know, David. What do you think? It's I think in general that similar systems are probably in place in many organs that require a balance between proliferation and but not having overproliferation. As David was showing, if you have too much of a good thing, you can have problems. And so it seems likely that most tissues will have these kinds of mechanisms built in, and they've probably evolved over time. They were selected for as positive for the system.
Yeah, I totally I agree with that. Like we see these patterns being repeated. Like they are um, these mechanisms are being used over and over in biology. And yeah, Sophia was pointed out the uh, thanks to these evolutionary pressures, they have been in place because they work.
Mm-hmm.
Yeah, I was thinking just it's of course way more complicated than that, but make a bunch of organoids of, brain, whatever, liver, all kinds of, and then pour you know, um, some common pollutants on it and see what happens. But this probably, I don't know, if you write a grant like this, people will say it's a fishing expedition, and then they will not give you a grant. I don't know.
Yeah, I think for sure. Well, from the grant perspective, yeah, I guess that would be one of the most reviewers would say that. I think that uh, so we let's say this example, this story that we just shared. It's we're looking at the cellular level, right? So, however, there are next level questions of what at least for example in the intestine but related to pollutants is like what the things that we're consuming nowadays well depending on the food source it can contain potential harmful um, carcinogens or harmful um, toxins and it this next level question is like how this might be affecting our microbiome, how this microbiome then is affecting at the cellular level in let's say in the context of DLG1 or high wind. So it's definitely this is humbly I humbly say it's just a tiny piece of a puzzle that that for sure it's part of a more complex um, uh, more complex context that is going on, and that's where also collaborations with other colleagues thinking about these problems in a from a different perspective. It's so valuable because it adds more pieces into the puzzle to hopefully get a better picture. But yeah, this is just a tiny bit uh, of a bigger picture for sure.
Hmm.
Yeah, so for example, in autism, it has been shown that migration of neurons during development is kind of faulty, and integration of the system is kind of not working well. So I was thinking of that, and um, but yeah, it would be interesting to kind of use this as a principle and then check other systems too. I think it would be really interesting. But I didn't want to take over the stage. I know Dr. Shah is here, Akil and Catherine. If you have questions, please go ahead. Thank you.
Yeah, thank you so much, both David and Afi, for your wonderful talk. My question in regards to the DLG1, and you mentioned about CDC42, and I was wondering because we have the same gene involved in a kind of cervical cancer by contribution of the HPV. And however, when we are talking about the mucosal immunity, it just make it more interesting. And I was wondering, maybe you have further information that you can share with us.
I don't know anything about cervical cancer. I don't know, David, if you've come across this in any of your reading with
I
CDC42.
have, yeah, no, I have not. The what comes across like after like during these reading for when putting together this story is that definitely, for example, CDC42 is it's this regulator of proliferation and migration, and it has been involved in EMT um, and cell migration during cancer. So that has been, but it has been in the context of other epithelial cancers. Regarding cervical cancer, I don't know. I know that. Um, I know that in certain contexts, it's also context dependent because it has been shown to be CDC42 increases promotes cell migration. However, in our studies, what we see is that an activation of CDC42 is having the opposite phenotype. And in talking with one of our collaborators, Natalie at McGill, she just before this paper came out, she had a paper showing that in other epithelial cancers, actually too much of CDC42 is detrimental for the survival of the cancer and for migration. So I think like even though we have, as I was saying though, that there are these patterns that are repeated in biology, it uh, the context matters.
As long as you just mentioned about IBD and polarity, somehow they are related to each other, and it can be, get involved into the DLG1 as well as CDC42. However, is there any specific reason that you just chose retroviruses? For example, why you didn't choose adenoviruses?
The you mean that for the infection, the rotavirus
Yes,
infection?
that was part of your slides.
So, yeah, well, the reason why we chose the rotavirus infection is that uh, our collaborator Mary Estes have at Baylor has uh, beautifully detailed how this infection takes place in the intestine and how it actually the virus doesn't affect the the creep compartment. So that was one of the reasons that we wanted this type of injury model. The second reason, and I think is well equally important, is that with rotavirus there is an increase in Wnt target genes and Wnt ligands in the intestinal niche as well. Well, in the intestinal mesenchyme and also in the intestinal epithelium. That is the 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 other element that we were looking for to have an injury system in which we have increased Wnt signaling in the creep compartment, and where the stem cells will be healthy. The only the only variable in that case was the loss of DLG1 in the epithelium. So I think that those two reasons were the main reasons why we chose this injury model.
And how many days you mentioned about the niche of the, I mean,
Yeah,
concentrated and neutrophil just gathering and all of those things? Do you have further
yeah,
explanation
no,
about that?
yeah, I actually didn't cover the specifics mainly because of time, but that is a great question. We gavage the mice with rotavirus, and it takes 4 days to have the peak of infection. And again, this has been nicely described by the SS lab. So what we did was we knocked out DLG1 prior infection, and then we infected with rotavirus. Let's say that was day 0, and we collected 4 days after infection. Since it has been since the SS lab previously reported that was the peak of infection.
I see. Thank you so much for your response.
Yeah, you're welcome.
Ophir, did you want to say something? Did you want to add something?
No, I think David did a great job of fielding all these questions, and he's really kind of the expert on the project. So thanks to David for doing that.
Yeah. Do you have time for 1 or 2 more questions? Or
Yeah.
yeah.
Yeah,
Okay.
yeah, of course.
Hi, Akil. How are you? Did you want to ask something?
Yeah, I had one question. So my brother, he suffers from like ulcerative colitis. So I have one question, like how might the manipulation of the DAC axis in the ISC be utilized to develop like novel therapeutic strategies for promoting tissue generation and repair in patients suffering from IBD such as Crohn's disease and ulcerative colitis. I mean, regeneration, not generation, regeneration.
Yeah, that's a great question. David, you want to go? You want me to answer it?
Go for it. Go for it.
So there's a big question in IBD about how much there's defects in regeneration versus we know that the main drivers are inflammation, but there's also an idea in the field that regeneration is defective, and so there's a lot of discussions about potential therapeutics that could fuel regeneration and improve healing. And the most obvious candidate for that is the Wnt pathway. And as we've talked about, titrating the levels of signaling is not trivial. And so I don't know that necessarily DLG1 itself would be a target, but by understanding all of the different components of the pathway and the ways that it can be regulated, we'll learn more that lays the foundation for development of therapeutics in the future. And David, you feel free to chime in on that.
And I completely agree. Like, understanding the Wnt pathway and DLG1 as we described as part of this pathway, that will help us on hopefully in the near future develop better therapeutics for Crohn's disease or ulcerative colitis for intestinal epithelial regeneration.
Yeah, I was thinking by studying the like interactions between the DAC axis and the gut microbiome to better understand like how the microbiome may influence the ISC behavior and Wnt signaling during the IBD, can potentially like open up like new avenues for the microbiome targeted therapies. So what do you guys think about this kind of strategy?
Over you want to
Yeah,
go or?
I'm not sure I quite understood. Can you tell me one more time what you're asking?
like one of the potential strategies, what I was thinking was like like by studying the interaction between the the DAC axis mentioned in the lecture and the gut microbiome to better understand how the microbiome may influence the ISC behavior and the Wnt signaling during the IBD conditions. Can potentially open up new avenues for
Yeah,
microbiome.
So no, I think I understand what you're asking. So the microbiome is clearly a very important part of a number of diseases, including in conditions like inflammatory bowel disease. And um, our lab and many other labs are looking at how the intersection between the microbiome and the inflammatory cells and the epithelium and other components of the system interact. And so it's probably not going to be the case that each one does its own thing, but rather that they're talking to each other, and that by manipulating one, you're going to manipulate others. So as David was mentioning, it's a complex system, but we think that slowly chipping away at it, we're making progress in terms of understanding the various components.
All right, yeah, that helps. Thank you so much for answering.
Yeah, I know Victoria joined later, so I don't know. I just wanted to give you a chance to speak, Victoria, if you don't have a question.
Oh, hi. No, I'm glad to be here. I'm sorry to arrive so late, and I'm appreciating everything that I'm seeing and hearing. Thank you very much.
Yeah, I had another question about how, or are you looking into like epigenetic mechanisms that might influence this axis of signaling, and which mechanisms might play a role during, especially during development and later on in aging, maybe.
David, you wanna answer that one?
Yeah, sure. Uh, so if I understood, Katarina, you're asking what's our take on epigenetic changes in the intestinal epithelium and how that also affects later in life. Is that
Yeah,
correct?
and how it affects this balance of then deciding between death or life, or and division and migration, like especially, yeah, I don't know, is there methylation or histone, or maybe even um G-force or I motifs going on due to different factors that then change RNA levels and gene expression.
Yes, like so we that is definitely a possibility. And at this point, we haven't looked at um, doing like, for example, an ATAC-seq on these intestinal stem cells. Looking back, well, hindsight is 2020, but so for sure it would have been nice to do a multi-omi approach with the isolated intestinal stem cells, but yeah, at this moment we haven't really looked at the the role of epigenetics in the context of DLG1 loss. Uh, but yeah, I don't know, Fear, if you had any speculations about it.
No, I think you've answered it well.
Yeah, thank you. Yeah, I think it's just an interesting field. I wrote some grants about looking, especially what interests me is these 3D formations, how they can change, because we talked about and in your model using, you introduced a virus, and I just thought it would be really interesting to see, to see these formations. I know there are antibodies that you can visualize these DNA 3D formations. Yeah, so I thought it would be interesting to look at it, but it would be a whole nother project and a lot of work. So
Yeah, definitely. It sounds quite interesting, but yeah.
it's always easy to have ideas, right? But to do all of it is the problem.
Agree.
And I know, Catherine, you have been trying to raise your hand. I invited you a bunch of times. It doesn't seem to work. So um, maybe do you want to share in the chat your comment or question, Catherine? Um, yeah, I'm really sorry about that, but it seems to not been working. Or maybe you don't want to raise the hand anymore. I just wanted to make sure you know that, I realized you want to speak and raise your hand. So yeah, I wanted to thank you for taking the time to come here and to share this really amazing work with us, and um, that the data is really interesting, and the fact that biology is again so fine-tuned, and that the context is really important, um, is also really interesting to hear. So yeah, thank you so much. We really appreciate it. And yeah, this was an amazing discussion. So
Thanks
I
for
hope you enjoyed it.
inviting. Yeah, thanks for inviting us.
Yeah, thanks so much for having us.
Yeah, wonderful. And yeah, maybe one day we'll speak again. We'll invite you again if you want to go through the hustle again to come here. And yeah, for everyone, thanks for asking questions and for being here. If you like discussions like this, you can join us again, of course. And the next talk will be actually about climate change and how reduced ocean CO2 uptake is going on due to slowing of circulation of ocean currents. It's a very different type of talk, but I think it will be also interesting. So thank you again, Ophir and David, and we wish you all the best for the future. It's really important work, and thank you so much again.
Thanks. It was
Thanks.
nice to be here.
Close the room in
Bye.
3, 2, 1. Bye, everyone.
Bye.
Bye.